Welcome to Labster. I am your assistant Dr. 1. Today, you will assess a scientific project using synthetic cells to see if they can help save the world. Let's get you set up for lab work so you can explore the lab to find out exactly why you are here. Find a lab coat and click on it to put it on. Find a glove box and click on it to put on the gloves. You can explore the lab by hovering over items in the lab to find out more about the science that happens here. Dr. Hacker will be with you momentarily.
Hi there, welcome to our synthetic biology lab. I am Dr. Bio Hacker and I see you have already met my AI assistant Dr. 1. We are trying to create synthetic cells that make useful compounds powered by photosynthesis. We already have a prototype of our synthetic cells, but we have some work to do to improve the health of the cells and the efficiency of the process. Just like real-life cells, our artificial cells contain a nucleus that holds the DNA, and this DNA is transcribed to RNA and then translated into proteins. The cells can even send signals to other cells by releasing proteins into the extracellular environment. Please join me at the hollow table to look at the cell membrane.
Activate the hollow table.
Remember that you can lower the lab pad to see the plasma membrane on the hollow table. In watery environments, lipids spontaneously form spherical structures that bury their hydrophobic tails and expose their hydrophilic heads to the water. It is this property that allows the membrane to make the cell a compartment separate from the extracellular environment. We even think that this is how the first cells formed. Isn't that fascinating?
Navigate through the plasma membrane using the hollow table buttons.
Can you see how different lipids have different colors on the hollow table? These different lipids, in combination with carbohydrates and proteins that are also found in the membrane, give the membrane some fluidity and elasticity. This is known as the fluid mosaic model.
Navigate through the plasma membrane using the hollow table buttons.
Many molecules need specialized transporter proteins to move in or out of the cell. This makes the membrane a selective barrier. In our synthetic cells, we have optimized the membrane composition, but the transport across the membrane needs a little more work. The cell membrane forms a compartment, or cell, that is separate from the extracellular environment. What is the other main function of the cell membrane? Great job! The cell membrane forms a selective barrier between the inside and the outside of the cell as it controls which molecules can move in and out of the cell. Due to the composition of the cell membrane, some molecules can cross the membrane more easily than others. This is called relative permeability. Which type of molecule is least able to cross the membrane without transporters? There is a poster in this room that will help you, or you can click View Image in the lab pad. You kept your eye on the prize. Ions carry a charge, which makes them hydrophilic. The inside of the lipid bilayer is hydrophobic and therefore does not allow ions through. Have you seen the poster on the wall in this room? It is a handy overview of the permeability of the plasma membrane to different types of molecules. You can also find it in the Media tab on the lab pad in case you need it later on.
Alright, let's head over to the bioreactor workbench. Grab the sample tube from the rack when you get there. The bioreactor contains a central compartment where the cells are growing, and a central processing unit that adds everything the cells need from various bottles. It also has a sample valve to take samples without contaminating the cells. Now, can you take a sample of the prototype cells from the central compartment of the bioreactor? Place your sample in the tube rack to dive into it to see a cell and its transporters up close. Teleporting to the virtual cell. On the right, you can see some common characteristics of molecules. Clicking on the button will launch that molecule at the cell. Select the type of molecule that can enter the cell without a transporter. If you are correct, you will unlock a transport protein on the cell that will allow you to transport other molecules into the cell. Nice job. As you can see, an aquaporin has now appeared in the cell membrane. Which types of molecules are transported by aquaporins? That's right. Aquaporins are channels in the membrane that allow water to pass through via osmosis. Aquaporins are unique among the transporters because they are always open. Other channel transporters and carrier transporters open and close, depending on the circumstances. Why don't you try it out by launching water at the cell? Nice job so far. You unlocked a channel protein. These proteins form a channel through the membrane that can open and close to allow specific molecules to cross. Select the type of molecule that might enter through a channel protein and launch it at the cell. You can even be creative and try to use two molecule characteristics together. Select a molecule using the buttons on the right to shoot it at the cell and determine whether it can cross the cell membrane. Good thinking, but this molecule has already entered the cell before. Try building a different molecule with the building blocks. Good choice. You unlocked a carrier protein. These proteins bind molecules on one side of the membrane and undergo a conformational change to release it on the other side. Select the type of molecule that might enter through a carrier protein to launch it at the cell. Which type of molecule would use a carrier protein to enter a cell? These channel and carrier proteins mainly transport molecules passively. Active transport, on the other hand, needs energy. Why does active transport require energy? Excellent. Passive transport is also called facilitated diffusion as it allows molecules to flow towards a lower concentration and/or down an electrical gradient. When molecules have to go against these gradients, it requires some form of energy. You are building transporters like a protein. You managed to find out that only small hydrophobic molecules can enter the cell without specialized transporters. Water enters the cell through aquaporins and bulky, polar or charged molecules need a channel or carrier protein transporter. Now let's look at some subtypes of these transporters and how they are controlled. You now have more types of transporters on the cell. Your goal is to choose the correct transporter for the type of molecule we are going to launch at the cell. You are now launching an ion. Which three transporters can let ions into a cell? Click on them. You've got this. Ion channels can be mechanically gated, voltage gated and ligand gated. I think you are ready to have another type of molecule and a few more transporters and mechanisms thrown into the mix. Now, you are launching 3 molecules that require carrier proteins to get into a cell. Click on the correct 3 transporter proteins for these molecules. Go you. Carrier transports can be uniporters, which only transport one molecule in one direction, symporters, which transport multiple cargoes in the same direction, or antiporters, which transport multiple cargoes in opposite directions. Let's add in another type of cargo molecule. Now, you are launching molecules traveling against a chemical gradient. Which three transporters could get these molecules into the cell? Awesome work! Transporting molecules against the electrochemical gradient requires energy. These molecules are transported by ATP-driven pumps, light-driven pumps or coupled transport, which uses the transport of a molecule traveling down the electrochemical gradient to drag another molecule against the electrochemical gradient. Which of these options is not a mechanism to control the opening and closing of ion channels? Excellent. Ion channels can be voltage, ligand, or mechanically gated. ATP is one of the drivers of active transport, along with light and coupled transport. How does coupled transport allow a molecule to be transported against the electrochemical gradient? That's right. Another molecule going down the electrochemical gradient drags the molecule of interest up the electrochemical gradient. This other molecule can either be traveling in the same direction in a symporter transporter or in the opposite direction in an antiporter transporter. You don't just build transporters, you know how to operate them too. Through these gating mechanisms, the cell controls which molecules go in and out of the cell. Let's go back to the lab to see if Dr. Hacker was able to use this information. Teleporting back to the lab. Wow, we sure learned a lot about transport proteins from the virtual cell. Dr. Hacker has used your learning from the virtual cell to insert transporter proteins into the membrane of some synthetic cells. Let's test if these proteins actually transport molecules across the membrane using fluorescence microscopy. Enter the lab. Great, let's start at the biosafety cabinet. For the next mission you need to use a pipette to transfer substances. Would you like to play through a short tutorial to learn how to use the pipette? The pipette is used to transfer very precise volumes from one container to another. Pick up the pipette. Click on the box with the pipette tips to pick one up. The disposable pipette tips are sterile plastic tubes. Make sure you only use a pipette tip once, otherwise you might contaminate your samples. Click on the water bottle to fill your pipette tip with water. In the real lab you would need to adjust the volume on your pipette. This is a bit tedious, so I will automatically set the correct volume and you don't need to worry about it in this simulation. Now transfer the water to the tube on the tube rack. You have successfully transferred the water. Now click on the trash bin to discard the used tip. Now you know how to use the pipette. There is a theory page about pipetting on your lab pad. You can access it at any time through the theory tab. We will use the pipette soon, but first, we need to find the cells. Put back the pipette on its holder if you are holding it. Dr. Hacker has already prepared the cells. You can find them in the incubator at the microscope workbench. You can open the incubator using the Open Close button with the arrows. Great. If you pick up the 6-well plate and take it to the biosafety cabinet, I will close the incubator. We will be using the fluorescence microscope. Can you figure out which substances from the biosafety cabinet you need to add to the cells to measure transport across the cell membrane? If you use the pipette to add the substances, I'll make sure to automatically set it to the correct volume. Add fluorescently labeled cargo molecules and the membrane dye to the cells in a well using the pipette. Remember to use clean tips. Did you manage to figure it out? Use the pipette to add the green cargo molecules and the red fluorescent dye to the cell plate. If you are unsure if you have added a substance to the cell plate, hover over the tooltip to hear what substances it contains. Remember to add a sterile tip to the pipette for each substance. You must dispose of the used tip in the trash bin before picking up a new one. Raise the lab pad after you have added both substances to the cell culture plate. Great! You are a real lab genius. Great job! You have prepared the sample correctly. It now contains the cells with a transporter protein, cargo molecules labeled in green, and a red dye to label the cell membrane. I will go ahead and prepare the rest of the samples for you. Why do we need to label the membrane? You are great at experimental design. The cell membrane is not fluorescent. Thanks to the membrane dye, we can see where the cells are. If the green signal from the cargo molecules overlaps with the red signal from the membrane, we know the molecules have entered the cell. Awesome! Now let's put the pipette back and take the cells to the incubator at the microscope workbench so the transporters can do their job. Open the incubator and place the 6-well plate inside. The cells need to incubate for an hour. Luckily, we are in a virtual lab and can speed up time. I will also wash the cells for you to remove any cargo molecules outside the cell. Wait for the incubation to finish. Let's take the cells to the fluorescence microscope. The fluorescence microscope uses lasers to excite different fluorophores. In our case, these are red attached to the membrane and green attached to the molecules. See if you can figure out whether the molecules have crossed the membrane. I have already selected the correct settings. You will see the results appearing on the screen next to the microscope, or by clicking View Image in the lab pad. Are the molecules able to cross the membrane? That's right. The green fluorescence inside the cells shows that the molecules entered the cells. As you can see, there is no green fluorescence outside of the cell. Clearly, all cargo molecules outside the cell after the incubation were washed off successfully. Dr. Hacker used a type of transporter called an ABC transporter from bacteria. Eukaryotes like humans also have ABC transporters. Since we are using synthetic cells here, we could use transporters from either system or modify transporters to create a new, synthetic version that does not naturally occur. Interestingly, while both prokaryotes and eukaryotes have ABC transporters, they are not exactly the same. The principle of membrane transport is the same across all the domains and kingdoms of life, but their transporters themselves vary. I'd love to explore this with you another time. Alright, let's go over to the wall screen now. Transport across the membrane can be highly specialized in differentiated cells. This allows the cell to execute its role and work together with other cells in the tissue or organ system it belongs to. On the wall screen, you will find transport proteins that are highly expressed in certain human cells or organs and their role. Let's see if you can match the cell type to the description. Pick up the organ card and add it to the empty space next to the matching description. Certain scorpions produce a toxin that inhibits the sodium channel closing. Now that you have identified the role of sodium channels in nerve cells, can you predict the effect of the scorpion toxin on neuronal function? Remember that you can lower the lab pad to take another look at the wall screen. Smart. If the sodium channel does not close properly, sodium ions leak into the cell. This makes the inside of the cell less negatively charged and lowers the threshold for depolarization. Great job! We have just seen how cells work together in organs and tissues. By transporting molecules across its membrane, each cell helps the organ system fulfill its role. Dr. Hacker will be so impressed. Let's go and find her in the lab room you first entered.
Ah, there you are. I have been following your progress and was actually able to implement some of your findings while I was working with the cells.
Take a look at the bioreactor screen. Do you remember what these stats looked like when you first arrived in the lab? If not, you can check out the Media tab. We are not producing a huge amount of insulin yet, but the cells are using more carbon dioxide, suggesting that the health of the cells and the amount of photosynthesis has improved.
Clearly, the health of the cells has improved. Compared to the situation when you entered the lab, how has adding the transporter proteins contributed to healthier cells? You got it. Before, the cell membrane was only a compartment that separated the inside of the cell from the extracellular environment. With the exception of small hydrophobic molecules, nothing could cross the membrane efficiently. Now that we have added various transporter proteins, molecules can move in and out of the cell. This means that the cell is now a selective barrier. Clearly, there are still lots of challenges for this project. For instance, I would like to increase the efficiency of photosynthesis. There are also lots of other applications for my synthetic cells that I would love to explore with you another time. Today though, your work on the cell membrane and transport has really helped us drive the project forward, and I hope you still think it can help change the world.
Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.